Thank you very much for giving me opportunity to talk to you this afternoon about ATM pathway in chronic lymphocytic leukemia. I have nothing to disclose. So ATM is a protein kinase that has both nuclear and extranuclear functions. And in nucleus, ATM acts as an active monomer. Upon its phosphorylation in response to DNA double-strand breaks, ATM synchronizes multiple cellular functions, and by virtue of phosphorylating a specific form of histone, has ability to influence chromatin structure that is amenable to DNA repair. And then sits at the decision what kind of DNA repair is going to be utilized. By phosphorylation of protein called 53BP1, ATM has ability to channel repair to a non-homologous end joining, and by its ability to phosphorylate BRCA1 protein, ATM facilitates homologous recombination repair, which operates upon S phase of cell cycle. And if DNA damage is excessive in cell, then ATM has ability to activate p53 and cell cycle checkpoints and induce programmed cell death or apoptosis. So it is obvious that one of the most important effects of loss of ATM function is propagation of cells with damaged DNA. In terms of extranuclear function, in terms of cytoplasmatic functions, ATM acts as an active dimer. And it has, again, multiple functions, but most importantly, it regulates level of reactive oxidative species and also regulates insulin signaling. And one of most important effects of loss of cytoplasmatic function of ATM is increased oxidative stress. So we know that ATM gene is localized on chromosome 11q23, which is frequently altered in chronic lymphocytic leukemia, both by virtue of deletion of this region, but also a number of mutations that can occur in this very large gene. And over the years, we've shown that these mutations can be both sporadic and acquired in patient germline, that they can serve as alternative to p53 mutation and inactivation, and that importantly they have phenotypic features such as accumulation of DNA damage, induction of a very specific transcription profile in response to DNA damage, and also importantly, ability to influence spontaneous progression of CLL, and both ourselves and others reported shorter time to first treatment in patients with ATM alterations. ATM mutations are particularly enriched in tumors in 11q deletion, where they lead to complete loss of ATM function. And it is very important to actually note that for the loss of ATM kinase activity, we need loss of both ATM alleles. And you can see here from this Western blot that if only one allele is altered by virtue of 11q deletion, a response is measured here as phosphorylation of downstream targets are completely retained. And the other important point is that in many patients, ATM mutations and 11q deletion are later inventing pathogenesis, meaning that they're subclonal. And therefore, indeed, even if we have loss of ATM function, it can be often attributed only to the proportion of tumor population, whereas the rest doesn't have these features. So for those patients that have inactivation of both ATM genes, both alleles, it is clear that a response and maintenance of a response to DNA damaging agents is altered. And we demonstrated this in 2012 in the context of CLL4 trial that you heard about this morning. If patients had inactivation of both alleles, they do as badly in terms of progression-free survival as patients that have both alterations of p53 gene. We are currently witnessing a huge successes in the management of CLL patients, but it is fair to say that even in an era of new treatments, very often we don't have optimal maintenance of responses in patients with 11q deletion that are enriched for loss of kinase activity. So one of the reasons for this being mentioned today, it's actually association of ATM with a large number of genetic changes in general with genomic instability. And we also know that ATM is associated with a very short, ATM loss is associated with very short telomeres in CLL, and the short telomeres are an appreciated factor in genomic instability. So there is quite a lot of reason to invent strategy that are selectively targeting ATM deficient CLL cells. And over the years, we focused on targeting both oxidative stress, but also we try to understand dependency DNA repair pathways that develop in CLL cells with ATM loss. So in terms of targeting oxidative stress, we have shown that both in vitro and in vivo, in context of xenograft models, we can induce very potent killing of ATM deficient cells if they're treated with pro-oxidants such as partenolide. And now I would like to spend a few moments going through various dependency pathways that can be targeted in ATM null cells. So one of the first dependency pathways that we targeted was PARP by utilizing a PARP inhibitor, olaparib. We observed synthetically lethal interaction between loss of ATM function and pharmacological inhibition of PARP, in which case PARP is single-strand break repair protein. Upon its pharmacological inhibition, a cell converts these breaks during replication of double-strand breaks, and there is an increased requirement for ATM function. So therefore, in ATM deficient CLL cells, we see tumor-specific killing, and we also see a very impressive response in vivo in xenograft models where olaparib leads to complete elimination of tumors. And what is important, that this killing doesn't require activation of p53 pathway, which in case of ATM null cells does not work anyway, but also leads to a dramatic accumulation of DNA damage that we can see here in a form of accumulation of gamma H2X foci. Encouraged by these findings, we opened some years ago a phase 1 clinical trial with PARP inhibitor olaparib for refractory CLL and other hematological malignancies where ATM defect is frequent. And we were able to show that olaparib was very well tolerated. And although this trial was focused on terminally ill patients, we were still able to show that if ATM pathway was inactivated, patients stayed longer on trial and were managed for a longer time. So the second dependency that I want to discuss with you is between ATM loss and pharmacological inhibition of principal responder to replication stress protein called ATR. If we pharmacologically inhibit ATR, then there is a requirement for role of both ATM and p53, and we were able to show that in case of ATM inactivation, there is a significant killing, specific killing of ATM deficient CLLs, both in vitro and in vivo in context of xenograft models. And interestingly, we also observed synergistic killing with ibrutinib by the virtue of the fact that ATR inhibitor and ibrutinib target different cellular populations. So this synergy is subject to a clinical trial that is currently ongoing in UK for refractory CLL. And in similarity to PARP inhibition, killing here is not is not involving p53 pathway, but rather dramatic effect on progression of replication forks, which become much shorter in ATM deficient and p53 deficient cells upon ATR inhibition. So the third dependency that is worth considering is between ATM defect and alterations in homologous recombination repair. And to demonstrate this interaction, we targeted a protein called deubiquitinase USP7. This deubiquitinase is highly expressed in CLL cells, in particular ATM mutant, compared to normal B cells. And pharmacological inhibition of USP7 has a potent effect both in vitro and in vivo in xenograft models, particularly when we use combination with chemotherapy that requires homologous recombination repair and induces it. So we provided a mechanistic explanation for such a synergy, and this is really the mechanism by which loss of USP7 alters homologous recombination repair. So I would like to now turn to dependency 4, and this is interaction between loss of ATM function and ribonucleotide excision repair, which is a reasonably new appreciated DNA repair pathway. And this DNA repair pathway resolves by far the most frequent type of DNA damage in replicating cells, presented here in these blue dots, which is erroneous incorporation of ribonucleotides during replication. It occurs more than over 1 million times per replication and is far more frequent than any type of DNA damage. And the principal responder to this type of alteration is RNase H2 complex that consists of 3 subunits. And we were recently able to show that one of these subunits, RNase H2B, localized on 13q deletion, is lost in a quite high proportion of CLL patients and leads to the functional loss of RNase H2 complex. And what is our newer finding is actually the fact that there is a synthetically lethal interaction between RNase H2 loss of function and ATM loss of function, and we can demonstrate this both in isogenic cell lines and in primary CLL tumors. And what I'm trying to say here, that actually RNase H2 can be therapeutic target for ATM deficient tumors, and we are currently busy developing inhibitor together with the CRUK drug development unit. So in the final part of my talk, I want to add another thought. So in addition to targeting oxidative stress and DNA repair dependency in ATM defective CLLs, perhaps we should think about interaction between DNA damage response and accumulation of DNA damage and immune evasion. Namely, what we are currently addressing is the fact that accumulation of DNA fragments in a cytosol of ATM deficient CLL cells is inducing cGAS-STING censoring pathway, which has ability to induce type 1 interferon response with a number of responsive genes, but one of which during long-term stimulation is PD-L1 molecule, and this can be involved, as you know, in evasion of immune response to tumor cells. So we are addressing possibility that particularly in this category of tumors, targeting this PD-L1 axis might be of some use. So I would like to make 3 conclusions to my talk. First, that ATM defective phenotype in CLL has both functional and clinical consequences. And due to genomic instability, ATM defective subclones may drive disease even in the era of new targeted treatments. And of course, this provides phenotype that can be targeted, definitely by targeting oxidative stress, various DNA repair dependency pathways, and potentially immune checkpoints in the future. I would like to thank various members of my groups, various internal and external collaborators, and of course our funders. And thank you very much for listening.
Thank you very much, Tanja, for your great talk. And yes, I see a question behind.
Hi, Matt Davids from Dana-Farber, in the back.
Yes.
You showed the data from Susan O'Brien showing shorter progression-free survival for relapse patients with deletion 11q on ibrutinib. But more recently, Tom Kipps just published a larger series with longer follow-up suggesting actually an improved progression-free survival with patients with 11q deletion. I'm wondering if you have any hypotheses as to why that might be.
I'm always a little bit suspicious, but when everything is measured just by 11q deletion, because this encompasses a number of tumors that don't have a functional loss of ATM. So unless we are addressing specifically tumors with functional loss of ATM, where clone is of sufficient size that we see a loss of ATM kinase activity, as I shown in the Western blot, I wouldn't like to really comment whether we are improving survival of ATM deficient CLL tumors.
I would like to ask you, considering all the findings you presented, do you think there is a need to introduce routinely analysis of ATM mutations in the diagnostics?
Well, I would probably most likely to see some functional tests which would identify a really subset of tumors where targeting is amenable. Otherwise, we are talking about a mixture of tumors with and without loss of ATM function. So this is worth consideration and careful thought how to approach it. Yeah.
So rather functional test than mutation analysis. Okay, thank you. As there are no more questions, thank you once more.